Hello my dear students and welcome back to my channel. In today's video, we will be discussing about the topic two-dimensional gel electrophoresis of proteins. This is a very important topic from the second chapter of your class 12 biotechnology. So, without wasting any more time, let us move towards the video. So, the topic for today's video is two-dimensional gel electrophoresis. So, first of all, what is electrophoresis? So, electro means electricity and phoresis means movement of molecules. So, if any molecule is moved in the influence of electricity, that is known as electrophoresis. This technique, the two-dimensional gel electrophoresis, was developed by the scientist O'Farrell and is used for the separation of proteins. Now, let us see the principle of this technique. As this process separates the proteins in two steps, so both the steps have different principle for separating the protein. The first step is isoelectric focusing, or we can say IEF. This process separates the protein on the basis of their isoelectric point. We will discuss about the isoelectric point later in this video. As the name of this technique is two-dimensional gel electrophoresis, so the first process, that is isoelectric focusing, is run in a horizontal way, and the second dimension or the second process, which is the SDS page, is run vertically, and the SDS page separates the protein on the basis of their molecular weight. We will discuss both these techniques in detail in the video. First of all, let us discuss the isoelectric focusing, or IEF. So, as already discussed before, that in isoelectric focusing, the proteins are separated on the basis of their isoelectric point, which is also known as pi. So, what is this pi, or the isoelectric point of the protein? So, the isoelectric point is the pH at which the protein has no net charge, means the protein has neither a positive or a negative charge. That pH is known as isoelectric point. So, let us understand this. Now, we all know that proteins are made up of amino acid, and generally these proteins exist as zwitterions. Now, what is a zwitterion? Let us see here. As the basic structure of an amino acid is that an amino group and a carboxylic group is attached to the chiral carbon. Now, this amino acid can exist in the zwitterionic form by the following reaction. The H plus from the carboxylic acid group comes towards the amino group and it forms NH3 plus and COO minus, means one positive and one negative charge. And as both the positive and negative charge are cancelled, so there is net zero charge. Okay, so these proteins exist as zwitterionic form, and when there is net zero charge on any protein, that pH is known as its isoelectric point, or the pi. Now, what happens when the pH is lesser than the pi value of the protein? So, when the pH is lesser, than the pi value of the protein has net positive charge. Whereas when the pH is more than the pi value of the protein has net negative charge on it. And due to this positive and negative charge, the proteins will move under the influence of the electric field. Now, this isoelectric focusing is performed on small IPG strips. IPG strips means immobilized pH gradient strips. These are thin strips where a pH gradient is set up with the help of ampholytes. Now, what are ampholytes? So, ampholytes are small molecules which have both the charges plus and minus on them, and they act as a pH gradient for the separation of the proteins. So, on the thin strips of gel, these ampholytes are immobilized and they migrate according to their pH and form a pH gradient. Like here is an example of an IPG strip where a pH gradient of 3 to 10 is set up. Means from the left side the pH is 3 and toward the right side the pH is 10, and this 3 to 10 pH is gradually increasing the pH. And when we will add the sample of the protein on this thin strip, the proteins will migrate in this pH gradient, and the pH at which the protein will have net zero charge, there the migration of the protein will stop and it will form a band. So, whatever proteins we have in our sample, all the proteins will migrate in the pH gradient strip, and wherever their charge becomes zero, they will stop migrating and form a band. And hence, the proteins get separated on the basis of their pi values. Now, this was the first dimension of the technique and it is run horizontally. Now, the second technique, which is the SDS page. So, what is this SDS page? So, SDS stands for sodium dodecyl sulfate, which is an anionic detergent. Anionic means it will have negative charges on it, and when we treat the protein samples with SDS, then this SDS gives overall negative charge to the proteins. And PAGE stands for polyacrylamide gel electrophoresis, means that in this case the gel is made up of polyacrylamide, and this SDS page is run vertically. So, this forms the second dimension of the technique. When we say two dimension, means that the first dimension is run horizontally and is placed after the first dimension is run, the gel is placed on the SDS page and then it is allowed to run vertically. When the SDS page is run vertically, then the protein's molecules will migrate according to their size, and the smaller protein molecules will migrate faster, and whereas the larger protein molecules will migrate slowly and will be found on the upper side of the gel. Now here you can see that after the first dimension or the IEF gel has run, the IPG strip is now placed on the SDS page and then allowed to run vertically. Now what happens in this case, that as we know that there are many different types of proteins present in our sample. So it could be possible that more than one protein would be having the same pi, means there would be one or two or three or some number of proteins which would have the same pi, and if they are having the same pi they will form a single band in the IPG strip. But it is not possible for a protein to have same pi and same molecular weight. So when the second dimension is run, then the proteins which are having the same pi, they will now migrate according to their molecular weight and form different spots. Whereas if we would only perform the SDS page, then there would be some proteins which would be having same molecular weight and they would form the same band. But due to the first dimension, as they would be having different pi values, they migrated at different bands. So, with the help of this technique, we can get a better separation of proteins, as two criteria are applied for their separation. The first is their pi value and the second is their molecular weight. This was all about the technique of two-dimensional gel electrophoresis. If at all you have any query, please do write in the comment section below. Thank you all for watching. Please do like and subscribe my channel.